this exercise, you're looking at the soil microbiology. It deals with exercise 50 and 51 from the lab manual. In exercise 50, it's looking at the genus Bacillus. These are gram-positive, as the name implies. They are bacillus or rod-shaped. They do form spores. They are commonly found in the soil, and they have a significant input and role in industry, agriculture, and medicine. And so in this lab, what you would be doing is you would take a soil sample, and then one aspect is to heat up water, mix the soil with water, heat that up. Because they are spore forming, what you're trying to do is to kill all the vegetative cells that are there, and then just have those spores that are remaining incubate, plate that soil out, incubate it, and then see if any of those spores do germinate. You could grow it on, plate it onto just a nutrient auger, and then you can do a multitude of tests once you've had it incubate for 24 hours. Number one, do a gram stain. If you see it's gram positive and it is rod shaped, you can do a spore stain if there are endospores. Then you want to continue doing additional tests such as a catalase test. You could do the starch test, the motility test. All of these biochemical tests would help you with the identification, is this in fact bacillus or not. As said, it has a significance in industry. Several of the enzymes that bacillus, certain species produce, are used in things such as adding enzymes in detergents to make them more effective. Some of the species do produce some antibiotics, which are used in terms of medical microbiology. In agriculture, some species have been used as biological pest management because they do attack and destroy certain other organisms such as fungi and insects. And then another aspect from medical microbiology is there is a species, Bacillus anthracis, which is the causative agent of anthrax, which of course is a health threat, and not just in general that it can be found in the soil, but also from use as a bioterrorist bioweapon. One of the other common soil microorganisms which is looked at is in exercise 51, and that is Clostridium. Clostridium is also a gram-positive. It is also bacillus-shaped. It also forms spores. So you might have to do some additional testing to determine, do I have the Bacillus genera or do I have Clostridium genera? One difference is Clostridium is anaerobic. And so when you try to isolate it from a soil sample, you need to grow the culture in a candle jar to reduce the oxygen levels so that you encourage the growth of this anaerobic organism. Several species of Clostridium have major roles in medical microbiology because they are pathogens. There is the particular species of Clostridium botulinum, which is the causative agent of botulism. You have Clostridium tetani, which is the causative agent of tetanus. You have Clostridium perfringens, which is a causative agent of gas gangrene, and then Clostridium difficile, which is commonly referred to as C. diff in the medical community. So there are several species that can cause some very serious illnesses, and so that is of concern because it is spore forming. Once again, once you have grown your cultures, you need to be very careful as you are doing the testing. You do a gram stain, do a spore stain, and then you'll want to do additional testing for specific for Clostridium to determine the species.